So okay guys, welcome. So we are discussing about the transportation types, right? So in active transportation, what happens is the so-called the movement of ions takes place against the gradient, and active transport takes place in the presence of ATP, right? In the presence of ATP, the transportation takes place, and it is also called as uphill transportation because the transportation of ions takes place from low to high concentration, that is low to high concentration. Okay, now that is one. Now if you ask me, there are some types of active transportation. There are actually some types of active transportation. Now the first one is primary active transportation, something called primary active transportation. It occurs due to or with the help of direct use of energy. Direct use of energy. Example, if you ask me the example, pumps, simple pumps. Which pumps? ATP pumps, right? ATP pump means these pumps which use ATP directly. So the primary active transportation are the ones which actually directly use the energy for transportation of the substances. Now if you take the second one itself called secondary active transport. Secondary active transport, it is actually coupled to the pump. How? For example, you take sodium symporter. Symporter means both the ions flowing in the same direction. Antiporter means ions moving in an opposite direction. So you take sodium symporter. We'll just we'll just delete this passive. We'll write completely regarding active only. You take sodium symporter with which ions? You take sodium amino acid symporter, or you take some more things like sodium glucose transporter. What happens is when the sodium goes in or out or into the cell, this transportation leads to generation or changes in the energy levels, helping the other ion to move with it. So that is why sodium symporters are regarded as secondary transporter, because there will not be active participation of the ATP. There will not be direct use of the ATP, that is direct use of the energy. Here what happens is the sodium transportation itself leads to generation or changes in the energy levels, causing the other ion to transport with it towards the same side. So that's why it's called sodium symporter. This also seen in sodium glucose transporter. Okay, so that is all about so-called active transport. Now regarding SgL2, I want to ask one question. Where is SgL2 seen and where is SgL2 seen? Anyone? SgL2 is seen in GIT. SgL2 is seen in kidneys, that is renal. Guys, please remember these positions or regions where these transporters, these all receptors present. Please don't forget. Okay, now we'll come to passive. So we already discussed there will be no participation of ATP. So it occurs in the absence of energy. Now it happens along the gradient. it happens along the gradient, but active transport occurs against the gradient, but passive occurs along the gradient. Okay, occurs along the gradient. So that's why it is called downhill. It is called downhill. Okay, now you can ask me, sir, from which concentration to which concentration it occurs? From high to low. High to low. High to low concentration. You got it? From high to low. For example, active generally occurs in, we'll discuss after a couple of times. So this is a high to low concentration exchange, that is passive transport. Now one of the most important statement that you should remember is passive transport follows Fick's law. Passive transport follows Fick's law. What is Fick's law? What is Fick's law? The rate of transportation increases with the increase in surface area of the membrane, decrease in thickness of membrane, and also decrease in size of molecules. So this is called Fick's law. The rate of concentration is increased by increase in surface area of membrane, decrease in thickness of membrane, and increase in size of the molecule. So passive diffusion follows Fick's law. Okay, that is very, very important. Okay, in passive diffusion we have some other types called simple diffusion and facilitated diffusion. Facilitated diffusion. Now what is simple diffusion? There will be no carrier. There will be no carrier. Example, anyone? Oxygen, carbon dioxide transport. But facilitated, carrier is present. Carrier is present, or carrier should be there. Okay, what is example? Example is glucose transporter. So remember, SGLT is a transporter which performs its function due to active process, but GLUT, glucose transporter, performs its function through passive process. Remember this difference. Okay, now we will discuss a very important topic, cell junctions. Guys, cell junctions are very important regarding dermatology. I am saying once again, dermatology, cell junctions are very important. Now if you ask me what are different cell junctions that we see, for example, there are different cell junctions. First one is gap junctions, second one is tight junctions, third one is desmosomes. Now regarding gap junctions, these are composed of connection proteins. Connection proteins. Now they help in transportation of ions, as well as the current means the voltage spread across from one cell to another with the help of gap junction. Now with the help of this statement, can you tell me one example for gap junction? Myocyte or cardiac cell, myocardial cell. Sorry, not myocyte. Sorry, myocardial cell. I did not write the complete statement. It's cardiocyte, or you can also call myocardial cell, cardiocyte. Why? The heart cell or the cardiac cell, they consist of something called gap junctions. You heard it right? Gap junctions present like this. Okay. These gap junctions are nothing but the cell membranes only of these successive cells. Okay. So these gap junctions helps in transportation of ions as well as spreading of voltage from one cell to another cell. And also you can see in so many examples like electrical synapses. Electrical synapses we already discussed, that is heart, smooth muscle, viscera, smooth muscles which are present in viscera like GIT, bladder, uterus, etc. And all of the fibers, you take it as smooth muscle or in the heart muscle, all of the fibers, they function as one unit called syncytium. Syncytium seen in gap junction. Now what are tight junctions? Tight junctions, they consist of something called jonula occludens. Further, they consist of jonula occludens, which are composed of occludin and claudin proteins. Occludin and claudin proteins. They are made up of occludin and claudin proteins. These tight junctions, they act as barriers, as simple as like that. Their main function is barrier. Now with this word, I think you can get the sufficient information into mind, like these are seen in blood brain barrier as well as testis barrier as well as placental barrier, kidney, etc. Now you understand, right? These blood brain barrier, testis barrier, placental barrier, they consist of this tight junctions. They act as barriers. See what a sweet thing. If you understand the simple components, now you can understand the higher components, like how drugs in pharmacology are prevented from entering into CNS brain. See, you will study some statements like it cannot cross the blood brain barrier, this drug cannot cross the blood brain barrier, this drug cannot cross the placental barrier, so it doesn't have any effect on the fetus. So these all things are completely based on physiological concepts only. If you have a strong physiological concept, then you can simply deduce the concepts, just like sweets, desserts, cakes. Okay. Now we discuss enough of it. Now we will discuss about the desmosomes, right? Desmosomes, they are also called as cellular cement, or you can also define it like macula adherens. Okay, macula adherens or cellular cement. These are made by adherin protein. These are composed of adherin protein as well as one more protein. What is that? Desmoglein protein. Okay. Now there are different types of proteins. I will tell you one short mini statement regarding dermatology. I think most of you might have got it, but still I will tell. DSG, desmoglein 1, desmoglein 3 defect leads to which condition? Yes, it's pemphigus. Okay, defect in desmoglein 1 and 3 in skin. 1 and 3 in skin leads to pemphigus. Okay. I think you are okay up to there. We will just draw a short diagram so that we can understand different cell junctions. Okay, now this is one cell. I will draw the worst drawing in the history. I will try to draw it as perfect as I can. You know, I am not a good drawer. Okay. I think in one series I have seen that dialogue. I have listened it. Sir, yes, he is a very good drawer, sir. Yes, the heroine just giggles after listening to that dialogue. Okay. Now this one, right? This one here, you see these are gap junctions, right? This is a cell. This is a cell. In between that, you have something called gap junctions. These are gap junctions. Okay. Now you have some other components we already discussed. What are these? Which have fibrils. We discuss desmoglein protein. Now you got it. What are these? Desmosomes. These are desmosomes. These are gap junctions. Okay. Now we have one more cell junctions, that is these are adherence junctions. These are adherence junctions. These filaments are called intermediate filaments. These filaments are active filaments. Okay, active filaments. Here, this is basal membrane or basal lamina. Remember, this is basal lamina. So here you will have focal adhesions. Here you will have focal adhesions. Okay, and you will also see other type of cell junction called hemidesmosomes. Hemidesmosomes. Now this is focal adhesions. These are hemidesmosomes, gap junctions, desmosomes, and these are adherence junctions. And here you can observe, I will show you one more type, that is tight junctions. These are tight junctions. Okay, enough of it. Now we will get on with, I think we finished the basic physiology, cellular physiology. We will just move with digestive tract or GIT. Okay, what is the time? I just want to check. It's 4:25. Okay, guys, physiology is not boring, but if you don't have correct explanation, then it will be boring. Okay, now first one, we should discuss some of the anatomical aspects. That means, what are the layers of GIT? There are four layers in GIT. What are those four layers? Mucosa, submucosa, muscularis and serosa. Mucosa, submucosa, muscularis and serosa. Okay, so these are the four layers, right? Now mucosa is the innermost layer. That means, what is meant by that? It forms the walls of the GIT, mucosa. Okay, and submucosa, we will first discuss about mucosa, right? It is the innermost layer and it contains something called villi. And microvilli for absorption. Villi and microvilli. What is their function for absorption? Now coming to submucosa, it forms the layers in vessels, right? nerves, lymphatics, glands. What is its main function? Secretion. Submucosa's main function is secretion. Remember, submucosa function is secretion. Okay. Mucosa function is absorption. Okay. Now muscularis. Muscularis is a muscle. Which muscle? Smooth muscle for motility. Muscularis layer is composed of smooth muscle which is useful for motility. Motility means movement. Okay. Serosa is for attachment. Why? Serosa is the outermost layer, so obviously it is for the attachment to adjacent components, right? But remember, serosa is absent in esophagus. Serosa is absent in esophagus, right? Serosa is absent in esophagus, known as adventitia. Just 1 minute, I will come again. 1 minute. Okay guys, so this layer, that is serosa layer, is absent in esophagus. Just remember this statement, called as adventitia. Okay, called as adventitia. So I think we will discuss about the diagrams in the latter part. We will just discuss the concepts, right? We will just discuss the concepts. So that's it up to the anatomical part. We will just go into the concepts. Now GIT is exclusively controlled by a system called, type of nervous system called enteric nervous system. Enteric nervous system, right? It actually controls the GIT, but it can be classified into two parts, not two parts actually, but you can classify it for further studying, right? What are those? First one is Meissner's. Do you have another name? Yes, you have submucous plexus. And second one is something called Auerbach's or myenteric plexus. Auerbach's or myenteric plexus. Now submucous plexus, name itself denotes that is present in the submucosa. It is present in the submucosa. So what is the function of submucosa? Mucosa is for absorption, submucosa is for secretion. So this Meissner's plexus or submucous plexus present in submucosa increases secretion. Now Auerbach's or myenteric plexus, that is present in muscularis. It is present in muscularis. Remember, Auerbach's plexus present in muscularis. Doctors, please don't confuse myenteric. Myenteric means muscle. Myenteric means muscle, remember myenteric muscle, so it's present in muscularis. Muscularis layer, what is the function? Contractility, that means motility. Motility means what does it do? What is its function? It increases movements. So myenteric plexus helps in increasing movements of the GIT, and submucous plexus or Meissner's plexus helps in increasing of secretion, right? So that is why if you know the physiology, you will know the pathology, you will know the medicine, everything. Okay. Now what about ANS control or autonomic nervous system control? Yes, there is also effect of autonomic nervous system on the GIT. Autonomic nervous system, that includes sympathetic, that is fight or flight. So what happens if sympathetic system activates? It decreases the GIT it decreases the GIT secretions, it increases the GIT secretion and as well as movements, right? The GIT secretion and movements. Also it increases heart rate, it increases reflexes, everything it increases except GIT, where the GIT secretions decreases and the movements also decreases if sympathetic system activates. What happens if parasympathetic system activates? Reverse of it, increases GIT secretions, increases GIT movements, everything it does. Okay. And previously we also discussed it increases reflex. Here urination also occurs, erection also occurs. Now you might get a doubt like, sir, what happens if sympathetic system activates? What happens to the excretion? What happens? Nothing. Sphincters gets closed, so defecation will not occur, right? Sphincters get closed, so you will be in a state of fight or flight thing, right? So defecation will not occur. Okay, so that is all about sympathetic and parasympathetic activation. Now how these systems activate, what receptors are present in those areas which leads to occurrence of these scenarios? M1 receptors are present in where? Stomach. M2 receptors are present in where? Heart. And M3 receptors are present in rest of the component, that is others. Okay, so remember these things, you always remember these things. Now if you know this basic physiology, now you know why, now you understand why the sympathetic drugs, parasympathetic drugs, now you can understand what is the basic principle behind the pharmacology. You understand, no? Parasympathetic drugs increase the GIT, they increase the movements. So where, in which condition you see decreased or paralyzed GIT movements? Paralytic ileus. Now can you give the drugs which initiate the parasympathetic system, or they accelerate or initiate the parasympathetic system? Can you give these drugs in paralytic ileus patient? Yes, you can give happily. Why? Because these parasympathetic system, if activated, it causes increase in GIT movements, as well it also leads to increase in GIT secretion. So paralytic ileus patient needs parasympathetic drugs. And what is the drug? Bethanicol. This is how you should study. This is how you should study. You take one action, you take one mechanism, you go to medicine, you go to pharma. One shot, two birds. Here three birds. What are those three birds? Physiology, medicine, pharma. Now you can even extract surgery from here. How? What is the abnormal condition of known due to abnormality in Auerbach's plexus? What is that condition called? Comment down in the comment section. What is the abnormal condition that is caused due to abnormality in Meissner's plexus? Comment down in the comment section. So many things you can get, not only one thing, right? So that is all about sympathetic and parasympathetic activation regarding GIT. Now you know GIT. Obviously GIT has nervous system protocol, it has some nervous system pathways, obviously, but who will start this nervous system pathway? Somebody should initiate, na? So that is called a pacemaker. Now who is the pacemaker of GIT? Somebody should start. Who is the pacemaker in heart? Sinoatrial node. Who is the secondary pacemaker in heart? Atrioventricular node, AV node. Now who is the pacemaker of GIT? Calss cells. It is not Kajal. It is not the beautiful Kajal you see in Magadera movie, or you see in some other movies like which movies you see Kajal? Which movies in Magadera movie you see Kajal? I think Magadera is my favorite. If you ask me in which movie Kajal is beautiful. So it is not Kajal, it is Kahal. It's not Kajal, it is Kahal, right? So it is not that Kajal, it is the pacemaker Kajal of GIT. What is the pacemaker of GIT called? Calls cells. Calls cells, not Kajal, Kahal. So you can remember like this: Kajal is the pacemaker. She will initiate some excitations in boys. By showing her beauty, she will bring excitation in boys. So Kajal is the pacemaker. Kajal is the pacemaker. Kajal initiates the excitation in boys. Boys means GIT. So Kajal is the pacemaker. Now Kajal is present where? Kajal is present in Auerbach's plexus. Auerbach's plexus also called myenteric plexus. Auerbach's plexus also called myenteric plexus. So these Calls cells, they make slow waves. They make slow waves. These also known as BER. What is meant by BER? Can anyone? Basal electrical rhythm. These Calls cells which are present in the Auerbach's plexus, they make slow waves called, they compose or initiate slow waves also known as basal electrical rhythm, right? Now when these slow waves plus stimulus, what is the stimulus here? Food. What is the stimulus here? Food or biryani. Food, distension, calcium, acetylcholine or GIT hormones, etc. These all are stimulus. These all can be taken as stimulus. You can take calcium, you can take GIT secretions, hormones, etc. Anything can be a stimulus. So when this slow waves plus stimulus when added, it leads to spike potentials. They cause spike potentials, right? Now this spike potential leads to binding of calcium to which component? Calmodulin. Calmodulin is a calcium binding protein. So when calcium binds to calmodulin, it leads to activation of myosin light chain kinase. You can call it shortcut MLCK. It activates myosin light chain kinase. Right, which will break ATP. Okay, this leads to contraction and movements of GIT. Now you understand? Now these Calss cells, which are the pacemakers of GIT, they produce slow waves, very small, slow, slight, light waves. These slow waves are called basal electrical rhythms. When they are paralleled, or when they are coordinated, or when they are added with a stimulus. What is that stimulus? It can be food, it can be calcium, it can be hormones, it can be GIT secretions, etc., etc. When this BAR is added with stimulus, then it leads to production of spike potentials. These spike potential leads to binding of calcium to calmodulin. What is a calmodulin? It is a calcium binding protein. So calcium binding to calmodulin leads to activation of myosin light chain kinase, MLCK, which will break ATP. Okay, this in turn leads to contraction and movements in the GIT. Now this is all okay. Now where in the GIT you will see maximum BAR, basal electrical rhythms? Duodenum. In duodenum. Now where in the GIT most of the basic drugs get absorbed? Second part of duodenum, second part of small intestine or duodenum. Now where in the GIT or where in the body most of the drugs get absorbed? It's duodenum. You understand? Guys, this is what physiology is. Okay, you know physiology, you can tell anything. The reason why duodenum is one of the part where maximum absorption of drugs takes place, right? Because it helps in production of maximum BAR, basal electrical rhythm. Now where the minimum BAR production takes place? Cecum is slow, very slow, right? Something like you take it in case of cecum, the production of BAR is like 2 per minute, whereas in duodenum the basal electrical rhythms are produced at a rate of 12 per minute. So that's the maximum, right? Okay, now we'll discuss something about GIT movements. 1 minute, guys. I think it's better to stop here. The time's up. It's 4:46, so we'll stop here. We'll continue tomorrow. I will make sure I will finish physiology in 3 days. Trust me, I'll finish physiology in 3 days. Trust me, just keep hope in me. And I just request all the students, one of my request to all the students is please you share and subscribe my channel. You just share the videos. If you want to share with your friends, it's your option, but subscribe my channel. Guys, in the upcoming days you are having one of the most beautiful good content, and trust me, we have good targets. IPL, Inicet Premier League, right? IPL, you know which subject I am concentrating. I am concentrating mostly on 4 subjects. Don't think like rest of the subjects I left. No. First one is medicine. Yes, most of the doctors say, see, medicine is vast. Don't read medicine. Don't waste your time in preparing for medicine. No. Medicine is simple if it is prepared by wise doctor. Your wise doctor has prepared the best of best material for medicine, so get ready. Second one is pathology. Pathology, standard material. Third one is surgery, get ready, surgery. And final fourth one, anyone can guess? SPM. So these 4 subjects are our best players. I marked SPM as Dhoni for our IPL team and surgery as Jadeja, medicine as Rohit Sharma, and pathology who? Kohli, King Kohli. And we have some cheerleaders also. Who is that cheerleaders? Can anyone? Who is that cheerleaders? Mehreen. Who is that Mehreen cheerleader? Forensic science. Right? And we have Anushka Sharma. For which one? Dermatology. And finally we have one more cheerleader. Who is that? Can anyone? Anyone guess? Who is that cheerleader? Okay, I will give you the subject. Which subject you guess? Who is that cheerleader? Okay, pharmacology. Can you guess which cheerleader I have allotted for pharmacology? Anyone? Okay, I will tell you, she is from Bollywood. Nora Fatehi. Okay, so I have just get all sort of practice ready for our IPL team. So make our IPL team as strong as possible. Okay, we will meet in the next class. Until then, revise what I have explained. Goodbye, Tata, your wise doctor. Thank you.